Hello everyone, welcome back to my channel. So, continuing with the series on biosystematics and taxonomy, today we will be going to discuss about a new topic, that is taxonomic procedures, and in that taxonomic collection, its preservations and the correct process of identification will be dealt with. So, taxonomy deals with the collection of the individual organisms. They are then classified, identified, and their proper preservation will take place, and later on, at last, publication is the result, that is its publication will happen. So collection of specimen is the vital but also a very difficult task for a taxonomist. That is, there are various methods that are involved in the collection of a particular organism. So let's see them step by step. So, collecting ways. There are various ways of collecting animals around the year. First is, these methods may be from picking a insect, flying towards a torch light, that is, as we know, torch or light attracts many insects toward it in the evening, or dredging for deep sea animals. Then the latter method requires elaborate and specifically designed equipments operated by a group of the sailors and scientists on an ocean-going vessels. That is, so much expertise is required if we are going to capture a sea animal or deep sea animal. Dredging means dredging is basically an instrument that is used to collect the water species. Then collecting is a highly specialized procedures in which a collector concentrate only on a group of organism. That is, each we can say a scientist or a collector should have to concentrate only on a single group of species and not on several. It will leads to disturbance in its work. That is the collection purpose. Then faunal surveys are carefully planned area-wise in a particular geographical area. One must be in the knowledge of all the possible geographical information of the place to be surveyed. That is, a collector should know the all possible geographical area whenever he must have to do that particular task. That is, whether hilly areas are there, presence of a water source is there, and other areas. Then including the distribution of various types of the vegetation, altitudes, and the season, etc. Then there are some areas in every country which are completely banned for ordinary citizens, that is, they can't go there and so cannot be surveyed to sample the fauna. So some areas are permitted to be surveyed and with special permission of a particular government, and involves many frustrating procedures also. So some countries have completely banned the collection of the animals by visitors. So a collector should have to consider all these characteristics before going to collect a particular organism. Then the first is the collection of insects. So insects form the most dominant group of animals due to their enormous number, along with combined with, we can say, diverse habits and habitat that they possess and that they reside in. So these are found in the air, in fresh or the brackish, that is salty water, shrubs, leaves or stem of a tree, low growing plants, plant products, seeds and fruits, debris, that is in the nest, in the soil, or in the insects or in other insects or the animals. So they have possess wide variety of habitats. By knowing their peculiar habit and the habitats, one can easily pick them by fingers. However, for quickly collection in the large numbers, special collecting instruments or the equipments are needed. So basically, first we have to understand the habitat and the nature of the insects. Then they are usually picked up whether by hands or by instruments. So methods of collecting insects. So insects can be collected using general nets during the sunny days. Light traps for the insects that are attracted towards the light are used. Example, malaise traps. So sweeping and beating are the most productive methods of collecting large number of insects. The principle of beating is to Hit a branch of a tree or a shrub by a heavy stick in such a way that insects and the other arthropods that are residing on that particular tree fall on the tray or the white sheet placed below from where they can be captured. That is, this is basically the simplest method utilized for capturing of insects. What we have to do is hit a branch of a tree, and then all the insects residing there on the tree will fall down and can be captured easily. So different types of aspirators are also quite useful in picking up the large number of small insects and other arthropods. So Burley's funnel or the flotation methods are useful in collecting small insects and other small arthropods inhabiting the soil. That is, those which resides in soil cannot be captured by the, we can say, beating method or sweeping method, debris, etc. So aquatic insects and other arthropods are best collected by dip nets or the plankton net. So this is a plankton net. It is used for capturing aquatic insects. Then this is a dip net, and this is another, we can say, net that is used for capturing the insects. Now the first instrument is insect net. That is, it consists of a wiring, a bag and a handle. As we have seen earlier in the diagram, the ring should be 12 to 15 inches in diameter and made up of about 3 mm iron or the steel wire. Then the ends of the wire are stout, which can fit into the grooves of the handle. That is, they should be perfectly fit into the handle. A fold ring is also quite easy to carry while traveling. So the bag is either of muslin cloth, fine nylon net or any other material depending on the method and the purpose of the collection. So this is the diagram of insect net. That is the handle that is made up of iron or steel, the wiring that is 12 to 15 in diameter, and that is the bag that is made up of whether muslin or other nylon net. Then another device is aspirator. That is, it is a simple suction apparatus used for collecting small insects and the arachnids. A vial of a glass or a transparent plastic is used. So now, what is a vial? So a vial is basically a small container, typically cylindrical and made of a glass and used especially for holding liquid medicines, etc. So it is utilized here. In one case, that is in figure 1 that we will be going to see in later stage, the vial is opened at one end, and in the other case it is opened at both ends. That is, here in this diagram the vial is opened at only one end, and in this case the vial is opened at both ends. Then two glass tubes pass through the stopper. The ends are provided with tightly fitting rubber stoppers, so to avoid the crushing of the small insects. That is, then a rubber tube is attached to the outer end of the suction tube for sucking purpose. The other end of this tube remains in the vial is covered with a piece of fine muslin cloth. A second glass tube is opened at both ends. So another type of aspirator is the bulb aspirator. It is very useful for the collection of mites, small insects and spiders. So these are the diagram representing different types of aspirator for collection of small insects and arachnids. This is the bulb aspirator. Then the vial is made up of transparent plastic or heavy glass whose both ends are tightly fitting with stoppers. So the one end of the intact tube is placing outside of the glass vial through which the entry of the insects is done. That is, as in this case, that is, from here insects will be entering, and then it here stopper is arranged or fitted so that they can't leave this aspirator via this method or this way. So the other end of the collection or the intact tube runs through the entire length of the vial and placed in the narrow hole of the other stoppers. So the suction is applied through the suction bowl and desired specimen is just in through the intact tube directly into the collecting vial containing 70% alcohol. So that's how an aspirator is used for collection of the insect specimens. Then the Burley's funnel. So it is quite useful in extracting insects and other small arthropods from organic soil and leaf litter. So it can also be used to extract the organism that is present in bark rooting, wood, fungi, mosses, flowers, stored food products, and the material from the nest of the birds, mammals, and the social insects. So Burley's funnel is basically used. So this is a diagram depicting the Burley's funnel. Here is the lid, the bulb. Here insect will enter the funnel and then it will lead to the container that will containing the 70% alcohol and triglycerin. Then it is the another bottle containing the required, that is ethyl acetate killing bottle. Then blotting paper is there, paper collection that is seated in ethyl acetate is there, and this is the cyanide bottle, that is the poison. Blotting paper is used, plaster of Paris, okay, and that is plaster of Paris dry and wet, both are used here, and this is the KCN solution, that is potassium cyanide. So apparatus of Burley's funnel is, it is a simple apparatus consisting of a metal or a plastic funnel having a wire mesh on its bottom for holding the sample. The narrow of the end of the funnel is present in a beaker, that is the container containing 70% alcohol and few drops of glycerin. Funnel is then covered with a lid having a hole in the middle for an electric bulb. As the upper part of the sample rises up due to the heat of the bulb, the organisms avoiding heat start migrating deeper into the funnel, and finally they are contained kept below in the container. So in this diagram, as the insects enter from here, so as to avoid the heat that is generated from the bulb, they avoid it and enters through this path, and by the way of funnel entering into the funnel, here a filter is utilized, and then they will enter into the container that is containing 70% alcohol. Then flotation method is also utilized for collection of insects. So this method is quite simple, and also to extract insect mites and other arthropods from the soil or the vegetation. It is also good for collecting eggs and the pupae of the insects from such materials. The sample is broken up is a basin containing a mixture of Mg2SO4 in the water in 1:3. Then after some time, the organisms start floating over the surface of the water, from where they are collected on a sieve or a filter paper. So this is the another method. So yeah, this is all about part 1 of the taxonomic procedures. In this video we have discussed about the collection methods of insects, and in another part we will be going to discuss about the collection methods of aquatic animals and the marine invertebrates, and also various preservation methods. So stay tuned for the next video. Hope you will like my video, and if you like my video, please do like, share and subscribe to my channel.